What if I told you that dinosaurs were not the first rulers of the Earth? That for 200 million years before them, the planet was dominated by other creatures, your direct ancestors, who already had saber-toothed fangs, who already had fur, who already had warm blood, and that these creatures almost became us, almost became the dominant intelligent animals on the planet, until the largest catastrophe in the history of life destroyed 95% of all species and reset everything they had been building for 100 million years? This is the story of the world before dinosaurs, the world that almost no one makes films about, because it ended too long ago and too terribly. But it is our world. We came from there. Before we go any further, please subscribe to this channel. Your support really helps us to continue our work. Thank you. There is a peculiar injustice in the way the history of life on Earth is told. The dinosaurs, which dominated the planet for approximately 180 million years, have become the defining emblem of prehistoric life. The creatures that appear in every children's book, every museum centerpiece, every major franchise about the deep past. And yet, the period that preceded them, the 350 million years that stretched from the first forests to the first dinosaur, was by almost any measure more extraordinary, more biologically creative, and more personally relevant to every person alive today. The world before dinosaurs is not a prelude to the main event. It is the main event for the story of what we are. The dinosaurs are the sequel. The story begins approximately 360 million years ago, when the Carboniferous period opened on a planet that was already ancient, but that was about to experience one of the most dramatic biological experiments in the history of life: the colonization of the land by large animals. The Devonian period that preceded the Carboniferous had seen the first amphibian pioneers make tentative forays onto the shore. But the Carboniferous was the period in which the terrestrial world truly became a biological frontier, the period in which forests, insects, and the ancestors of all land vertebrates underwent the explosive diversification that would set the trajectory of life on land for the next 300 million years. The Carboniferous world would be unrecognizable to any modern observer, and not merely because its fauna was strange. The oxygen content of the Carboniferous atmosphere was dramatically higher than today, reaching approximately 35% of the atmosphere compared to the modern 21%. This elevated oxygen level was the direct consequence of the extraordinary productivity of the Carboniferous forests. The vast coal swamps dominated by enormous lycopsid trees like lepidodendron and sigillaria, reaching heights of 50 meters, and by the giant horsetail calamites, which grew to 20 meters. These forests were producing oxygen through photosynthesis at a rate that outpaced the rate at which oxygen was being consumed by biological respiration and geological oxidation, progressively enriching the atmosphere with a gas that would have profound consequences for the size and biology of the animals living within it. The most immediate and most visually dramatic consequence of the elevated Carboniferous oxygen was gigantism in insects and other arthropods. In modern conditions, insects and other arthropods breathe through a passive diffusion system, a network of tubes called tracheae that carry oxygen from the body surface to the tissues. This system works well for small animals, but becomes inefficient at larger body sizes because the diffusion distance from the tracheal openings to the innermost tissues becomes too great for adequate oxygen delivery. The high atmospheric oxygen of the Carboniferous dramatically extended the size range over which this passive diffusion system could function effectively, allowing arthropods to reach body sizes that are simply impossible in the modern atmosphere. Meganeura moni was a griffin fly, a member of an extinct group related to modern dragonflies, that achieved a wingspan of approximately 70 centimeters. 70 centimeters from wingtip to wingtip, the size of a modern seagull flying on insect wings, in an atmosphere that smelled of ozone and oxygen. It was the largest flying insect that has ever lived, and it was not an exceptional freak, but a member of a diverse group of giant flying insects that patrolled the Carboniferous forest canopy. Its eyes were enormous compound structures providing near-spherical vision. Its mouthparts were designed for grasping and dismembering prey, and it was the unambiguous aerial predator of the Carboniferous sky, a role that would not be challenged until the pterosaurs evolved more than 100 million years later. Arthropleura was even more impressive, a relative of modern millipedes. Arthropleura reached lengths of approximately 2.5 meters, the longest terrestrial arthropod that has ever lived, roughly the length of a modern saltwater crocodile. Fossil trackways of Arthropleura have been found in Carboniferous sediments in Scotland, the trackways showing an animal of considerable width moving across the substrate with a characteristic multiple-legged gait, the impressions of its legs preserved in the ancient mud. These trackways are among the most dramatic physical records of the Carboniferous world. Direct evidence of a meter-wide, multi-legged giant moving through the coal forest undergrowth. Recent research published in 2021 and cited through 2025 found an exceptionally well-preserved Arthropleura specimen in Northumberland, England, confirming the animal's impressive scale and providing the first direct evidence of what it ate: plant material, including spores and fern fragments, establishing that the largest arthropod ever to walk the land was a herbivore. The Carboniferous was also the period in which the most consequential split in the history of vertebrate life occurred. Among the early amphibians that had colonized the land, a group evolved a revolutionary biological innovation: the amniote egg. The amniote egg, the kind of egg that a modern chicken lays, or a snake or a lizard, is a self-contained life support system. It contains not only the embryo, but also a membrane-enclosed fluid sac that serves as the embryonic ocean, a nutrient supply in the form of a yolk, a waste container, and an outer shell or membrane that allows gas exchange while preventing desiccation. The amniote egg liberated its producers from the absolute dependence on water for reproduction that amphibians retained, and in doing so, it opened the entire terrestrial world, the dry uplands, the seasonal environments, the continental interiors, to vertebrate colonization. The first amniotes were small, lizard-like animals, creatures like Hylonomus lyelli, known from fossils found inside the hollow stumps of Carboniferous sigillaria trees in Nova Scotia, Canada. that would not have attracted much attention from any observer who did not know what they represented. But what they represented was the beginning of all subsequent terrestrial vertebrate life. From the amniote ancestor, two fundamental lineages diverged, distinguished by differences in the arrangement of openings in the skull behind the eye socket. The sauropsids, whose skull had two temporal openings or none, and the synapsids, whose skull had a single temporal opening. The sauropsids would eventually produce the reptiles, the dinosaurs, and the birds. The synapsids would eventually produce the mammals, and you. The early synapsids of the Carboniferous and early Permian, the paleontologians, are the first actors in the story that leads directly to humanity. Paleontologians were the dominant large vertebrates of the early Permian world, occupying ecological roles from large herbivores to large carnivores across the landscapes of what was becoming the supercontinent Pangaea. They looked, superficially, like large lizards. They moved with a sprawling gait, their limbs extending horizontally from the body rather than being held vertically beneath it, as in modern mammals. Their biology was in many respects intermediate between the cold-blooded ectothermy of modern reptiles and the warm-blooded endothermy of modern mammals, a biological middle ground that is no longer occupied by any living animal and that can only be inferred from the fossil record. The most famous pelycosaur is also one of the most misidentified animals in the history of paleontology. Dimetrodon, the sail-backed predator of the early Permian, known to every child from toy sets and illustrated books, is routinely classified in popular culture as a dinosaur. It was not a dinosaur. It was not remotely related to dinosaurs. Dimetrodon is a synapsid, a member of the lineage that leads directly to modern mammals. It predates the first dinosaur by approximately 40 million years. If you must compare it to anything living today, compare it to you, not to a Tyrannosaurus. The temporal opening in its skull, the structure of its teeth, the metabolic hints preserved in its bones. All of these place Dimetrodon firmly in the ancestry of mammals and firmly outside the ancestry of any reptile or dinosaur that ever lived. Dimetrodon was a formidable animal. Adults reached lengths of approximately 3.5 meters and are estimated to have weighed up to 300 kilograms. Its skull was deep and powerful, equipped with both large canine-like stabbing teeth and smaller shearing teeth. The first vertebrate animal to evolve differentiated dentition of this kind, a tooth pattern that modern mammals have inherited and elaborated. This differentiation of tooth types, incisors for cutting, canines for stabbing, premolars and molars for processing, is one of the defining characteristics of the mammalian skull, and it appears here, for the first time, in a 300-million-year-old synapsid that children call a dinosaur. The sail on Dimetrodon's back, the structure formed by enormously elongated vertebral spines covered in life by a membrane of skin and blood vessels, is the most discussed morphological feature of any Permian animal and remains one of the most debated structures in the history of paleontology. The hypothesis most widely accepted today is thermoregulation. The sail, turned perpendicular to the sun, could absorb solar radiation and warm the animal's blood more rapidly than a sailless body of the same mass. giving Dimetrodon a morning thermal advantage over competitors that had to wait longer for their muscles to reach optimal operating temperature. Research published in Science News in 2026 on the nocturnal behavior of early synapsids. Based on the study of scleral ossicles, the small ring-shaped bones embedded in the eyes of many vertebrates, whose dimensions reflect the daily activity pattern of the animal, suggests that some early synapsids were already adapted for low-light or nocturnal activity, which would have made rapid morning warming through a solar sail particularly advantageous. But the paleontologists, for all their biological interest, were only the opening act. The Middle Permian saw the paleontologists give way to a new and more sophisticated group of synapsids, the therapsids. The therapsid radiation, beginning approximately 270 million years ago and lasting until the end of the Permian, produced the most biologically complex and ecologically diverse group of non-mammalian vertebrates that has ever existed. The therapsids were not merely more advanced than the paleontologists in some abstract taxonomic sense. They were moving toward mammalhood in multiple biological dimensions simultaneously, developing the posture, the metabolism, the skull structure, the dentition, and possibly the integument of modern mammals over the course of approximately 30 million years of rapid evolution. The therapsids divided into several major groups, each occupying different ecological roles, and each representing a different experiment in the approach to mammalian biology. The cynodonts were the dominant herbivores of the Permian world, stocky, barrel-bodied animals ranging in size from small burrowing forms to large hippopotamus-like browsers. Their skull was unusual. Most of the teeth were replaced by a tortoise-like beak of keratin, with the exception in most species of a single pair of enlarged tusks in the upper jaw. The purpose of the tusks varied by species. Some were used for digging, others for combat, others for display. But the overall body plan was extraordinarily successful. Cynodonts were the most abundant large herbivores on the Permian landscape, their fossils found on every continent, their diversity spanning dozens of genera over tens of millions of years. Among the carnivorous therapsids, the gorgonopsians occupy the most dramatic position in the Permian ecological hierarchy. Gorgonopsians were large, active, predatory synapsids with skulls that combined the ancient architecture of the earliest amniotes with a suite of predatory adaptations that were, in some respects, more advanced than anything seen in early dinosaurs. The skull of a large gorgonopsian was deep and powerful, with enlarged temporal openings that accommodated large jaw muscles. Muscles that produced the closing force needed to deploy the most distinctive feature of the gorgonopsian skull, the enlarged canine teeth. The upper canines of large gorgonopsian species projected downward from the upper jaw to an extent that rivals the saber teeth of the Pleistocene felid Smilodon. They are saber teeth, appearing in the synapsid lineage 200 million years before Smilodon, evolving independently the same solution to the same predatory problem. The largest known gorgonopsian is Inostrancavia alexandri, known from fossils from the Dvina River region of Russia. Inostrancavia was a substantial animal. Estimated body lengths range from 2 to 3 meters in different specimens, with skull lengths of up to 45 centimeters. The skull's canines were enormous, projecting approximately 15 centimeters below the lower jawline when the mouth was closed. The locomotion of Inostrancevia was more erect than that of the pelycosaurs. The hind limbs were held more directly beneath the body, in a posture intermediate between the full sprawl of modern lizards and the fully erect posture of modern mammals. This more erect posture allowed more efficient locomotion and, probably, the sustained activity that distinguishes endothermic mammals from ectothermic reptiles. Whether gorgonopsians were genuinely endothermic, whether they generated body heat internally through elevated metabolic activity, is a question that cannot be answered definitively from their fossils. The bone texture of gorgonopsians, examined by researchers using histological sections, shows a fibrolamellar structure that in modern animals is associated with rapid growth and high metabolism. Some gorgonopsian species show evidence of a secondary palate, a bony shelf separating the nasal passage from the oral cavity that allows an animal to breathe and eat simultaneously, a feature associated with the elevated food requirements of warm-blooded physiology. The exact point in therapsid evolution at which true endothermy was established remains debated, but the evidence increasingly suggests that the transition was occurring during the Permian, well before the first true mammals appeared in the Triassic. The Therocephalians, another major therapsid group, were in many respects even more mammal-like than the gorgonopsians. Their skulls show a greater reduction of the bones of the lower jaw, with the dentary bone, the tooth-bearing bone that forms the entire lower jaw in modern mammals, becoming progressively more dominant. Their teeth show complex differentiation between incisors, canines, and post-canine teeth. Some Therocephalian species show evidence of facial whisker pits in the snout bone, the nerve foramina that in modern mammals mark the attachment points of vibrissae, the sensory whiskers that are a characteristic mammalian feature. If the whisker interpretation is correct, some Therocephalians were furry, whiskered, warm-blooded predators hunting in the Permian landscape 30 million years before the first dinosaur left its first footprint. The cynodonts, the therapsid group from which true mammals ultimately descended, appear in the fossil record in the late Permian and diversify extensively in the Triassic. The name means dog teeth, reflecting the elaborate differentiation of the cynodont dentition that represents the most complete approach to mammalian tooth structure yet achieved in a non-mammalian vertebrate. The skull of a late cynodont is, in structural terms, almost a mammal skull. The dentary bone has become so large that it dwarfs all the other lower jaw bones. The temporal opening has enlarged to accommodate the large temporal and masseter jaw muscles. The secondary palate is complete, but the cynodont is not yet a mammal. It still has multiple bones in the lower jaw and lacks the fully mammalian middle ear that defines the class. These extra lower jaw bones are among the most instructive transitional structures in the history of vertebrate evolution. In cynodonts, they form the jaw joint, but in mammals they have migrated from the jaw into the middle ear, becoming the malleus and incus, two of the three tiny bones that transmit sound vibrations in the mammalian ear. The bones in your ears were once your jaw. The world in which all of these extraordinary creatures lived, the late Permian world of Pangaea, was itself extraordinary in its geography and climate. Pangaea, the supercontinent that assembled from the collision of smaller Paleozoic continents. through the Carboniferous and early Permian, was a landmass of remarkable scale, a single piece of continental crust extending from the North Pole to the South Pole, wider at tropical latitudes than the distance between the Pacific and Atlantic coasts of modern South America and Africa combined. The interior of Pangaea was, by the mid-Permian, an extreme continental environment, hot and dry in summer, cold in winter, with seasonal swings that had no modern analogue and that challenged the thermoregulatory systems of every organism that lived there. The coasts of Pangaea and the Tethys Sea, the broad east-facing ocean embayment that separated the northern Laurasian portion of Pangaea from the southern Gondwanan portion, supported more temperate conditions, and the marine fauna of the Permian Tethys was, at its peak, among the most diverse in the Paleozoic. Coral reefs of remarkable complexity lined the Tethyan shores. Foraminifera called fusulinids built carbonate skeletons, the size of grains of rice that accumulated on the seafloor in layers meters thick. Ammonoids, the coiled cephalopods that would go on to diversify so spectacularly in the Mesozoic, were present in the Permian Ocean in considerable diversity. The brachiopods, which had dominated the Paleozoic seafloor since the Cambrian, were at or near their peak generic diversity. And the crinoids, the sea lilies whose fossil remains give the Carboniferous limestone of England and the Permian reef complexes of West Texas their distinctive character, filtered the current with feathery arms in communities of extraordinary abundance. Then, approximately 252 million years ago, everything ended. The end Permian mass extinction is not the most famous extinction event in the history of life. That distinction belongs to the end Cretaceous event that killed the non-avian dinosaurs. But it is by every objective measure the most catastrophic. The Cretaceous Paleogene extinction eliminated approximately 76% of all species. The end Permian extinction eliminated approximately 96% of all marine species and 70% of all terrestrial species. It is not a quantitative difference of degree. It is a qualitative difference of kind. The end Cretaceous extinction was a catastrophic pruning. The end Permian extinction was almost total annihilation. The cause of the end Permian extinction is now understood with considerable confidence, though the details continue to be refined by ongoing research. The primary driver was the eruption of the Siberian Traps, a large igneous province in what is now Siberia that erupted approximately 3 million cubic kilometers of basaltic lava and, more critically, injected enormous volumes of carbon dioxide, sulfur dioxide, and other volcanic gases into the atmosphere and ocean over a period of approximately 1 to 2 million years. The carbon dioxide drove rapid and severe global warming. Estimates of the temperature increase at the extinction horizon range from approximately 5 to 10 degrees Celsius in global average terms, with larger increases at lower latitudes. The sulfur dioxide drove acid rain that devastated terrestrial plant communities. The warming of the ocean drove the dissolution of oxygen in seawater. Warm water holds less dissolved oxygen than cold water, producing global marine anoxia that spread across the seafloor and into the shallow coastal environments where most marine life lived. But the Siberian Traps lava flows did not kill 96% of marine life directly. The eruptions triggered a cascade of feedbacks that amplified the initial perturbation far beyond anything that the volcanic activity alone could have produced. The warming of the permafrost in Siberia released stored methane, a greenhouse gas many times more potent than carbon dioxide, that further accelerated the warming. The acidification of the ocean disrupted the calcification of carbonate-shelled organisms, weakening and killing the corals, brachiopods, and foraminifera that built the reef ecosystems of the Permian Sea. The collapse of marine productivity reduced the oxygen consumption of decomposing organic matter in the surface ocean, further depleting deep-water oxygen. And the elevated temperatures drove the expansion of the oxygen minimum zone, the naturally occurring layer of low-oxygen water that exists in all modern oceans, until it covered not just the deep sea floor, but the shallow continental shelves where the richest marine communities lived. The pace of the extinction was, by geological standards, terrifyingly fast. The most precise dating of the end Permian extinction, published by researchers using high-resolution uranium-lead dating of volcanic ash layers interbedded with the extinction horizon, places the main extinction pulse within approximately 60,000 years, a geological instant that in biological terms corresponds to roughly 3,000 generations of a large animal. The coral reefs and brachiopod communities that had taken hundreds of millions of years to reach their Permian peak of complexity were eliminated within a span that, for comparison, is roughly 6 times the duration of recorded human history. To understand what the end Permian extinction actually looked like as an experience, as opposed to a set of statistics, requires reconstructing it from the combined evidence of the geological record, the chemical record preserved in the rocks at the extinction horizon, and the biological record of what lived before and what survived after. The first signal would have been the acid rain. The sulfur dioxide injected into the atmosphere by the Siberian Traps eruptions reacted with atmospheric water vapor to form sulfuric acid, which fell on the forests and soils of Pangaea with a destructive chemistry that burned the leaves of the Permian conifers, acidified the rivers and lakes, and leached the nutrients from the soils. This was not the dilute acidification of modern industrial acid rain. It was concentrated enough in the worst periods of volcanic outgassing to strip the bark from trees and kill vegetation across continental scale areas. The coal gap that defines the early Triassic, the near-complete absence of coal-forming swamp forests, is partly a record of this acid rain destruction. Then the warming began in earnest. The carbon dioxide released by the Siberian Traps eruptions dissolved in the ocean, acidifying it, a process that attacks the calcium carbonate shells of marine organisms and makes it increasingly difficult for corals, brachiopods, and foraminifera to secrete the skeletons on which their lives depend. The reef organisms that had been building carbonate structures for hundreds of millions of years found the chemistry of the ocean shifting against them. The pH of the Permian Ocean at the extinction horizon, reconstructed from the isotopic composition of carbonate fossils, dropped by an estimated 0.7 pH units, a change comparable in magnitude to the worst-case scenarios projected for the modern ocean by the end of this century under high-emission trajectories. The reef corals and sponges bleached and died as their symbiotic photosynthetic organisms abandoned the warming, acidified water. The fusulinid foraminifera, those rice grain-sized architects of the Permian seafloor, dissolved as the carbonate saturation of the ocean fell below the threshold at which their shells could be maintained. In the water column, the expanding oxygen minimum zone rose from the deep ocean toward the surface, pushing the boundary between oxygenated and anoxic water progressively shallower until it intersected with the continental shelf environments where the richest marine communities lived. Fish that had always inhabited well-oxygenated coastal waters found themselves in water that could not support their respiration. The last ammonoids and nautiloids of the Permian Ocean retreated toward the surface, following the last thin layer of oxygenated water as the anoxic zone expanded beneath them. The sea floor of the shallow Tethys Sea, which in the mid-Permian had been carpeted with brachiopods, crinoids, and bryozoans in communities of extraordinary diversity, became a black, sulfurous, anoxic mud in which nothing could live. On land, the warming was even more extreme in its consequences. The interior of Pangaea, already the hottest and driest part of a large supercontinent, experienced temperature increases that pushed the equatorial regions past the thermal tolerance limits of almost every organism that lived there. Research using paleotemperature proxies from Permian-Triassic boundary sections in South China, the most complete continuous sedimentary record of the extinction horizon available, suggests that tropical ocean surface temperatures at the extinction peak reached approximately 40 degrees Celsius. Temperatures at which most animal protein begins to denature, and at which photosynthesis becomes thermodynamically impossible for most plant species. The tropical zone of Pangaea became, for thousands of years, genuinely uninhabitable, a thermal dead zone in which no vertebrate could survive, and in which plant communities were essentially absent. The gorgonopsians died first among the large therapsid predators. Their metabolism, probably approaching mammalian levels of endothermy, required large quantities of food, and the collapse of the Permian vegetation and the consequent collapse of the dicynodont prey base left them with nothing to eat. The great saber-toothed hunters of the late Permian simply ran out of prey. Their bones disappear from the fossil record at or just below the extinction horizon in every continental sequence where they have been studied. The dicynodonts lasted longer. Their herbivore metabolism required less food per unit body weight than the gorgonopsian predators, and the last surviving dicynodont lineages persisted through the worst of the extinction, with Lystrosaurus eventually emerging as the dominant vertebrate of the earliest Triassic. But the diversity that had characterized the Permian dicynodont community was gone. The dozens of genera that had occupied the full range of herbivore niches were reduced to a single lineage, the biological equivalent of reducing all the world's ungulate diversity to a single species of pig. The cynodonts that carried the mammalian lineage through the extinction were, to all appearances, among the least impressive organisms in the late Permian community. Small, probably nocturnal, insectivorous or omnivorous in diet, burrowing in habit. These are the biological characteristics that the cynodont survivors of the Permian extinction appear to have possessed, reconstructed from their tooth-wear patterns, their skull anatomy, and the scleral ossicle evidence for nocturnal activity. They were small enough that the collapse of the large plant community did not eliminate their food supply. Insects and small invertebrates persisted through the extinction in greater numbers than large vertebrates. They were burrowing enough to find refuge from the extreme surface temperatures. They were flexible enough in their diet to exploit whatever food sources the devastated early Triassic landscape offered. The world they emerged into was almost unrecognizable from the world their parents had inhabited. The Permian reef communities were gone. The diconodont herds that had carpeted the Pangaean uplands were reduced to Lystrosaurus. The gorgonopsian predators that had shaped the evolution of every prey animal in the late Permian ecosystem had vanished completely. The forests that had provided the structural framework of the terrestrial ecosystem were gone, replaced by opportunistic fern communities and simple shrub vegetation growing in soils still damaged by acid deposition. The early Triassic world was genuinely impoverished, not just in the number of species, but in ecological complexity, in the diversity of biological interactions, in the accumulated evolutionary achievement that makes a mature ecosystem more than the sum of its component species. It was, in the most precise geological sense, a reset, not an ending. Life had survived. The cynodont and dicynodont lineages were intact. The archosaur lineage that would produce the dinosaurs was present in its early forms. But a erasure of almost everything that had been built in the 100 million years preceding the catastrophe. The Permian Triassic boundary is not just a line in the rock. It is the boundary between two different worlds. The synapsid world that had been building toward mammalhood for 100 million years, and the archosaur world that would spend the next 200 million years building toward the moment when the mammals would finally inherit what their ancestors had almost achieved before the Siberian Traps erupted and took everything away. The terrestrial extinction was the same story told in different animals. Everything described in the preceding pages, the gorgonopsians, the dicynodont diversity, the ecological complexity of the late Permian, was gone. What remained was Lystrosaurus. But before we follow the story to its catastrophic end, we need to spend more time in the world that was lost, because it deserves to be seen, not merely catalogued. The Carboniferous coal forest was not a jungle in any modern sense. It was something stranger and more alien than any tropical rainforest alive today. The dominant trees, the lycopsids, did not grow in the way that modern trees grow. Lepidodendron, the most abundant of them, reached heights of 50 meters on a trunk that was barely 1 meter in diameter at the base, tapering to a forked crown of grass-like leaves. Its bark was covered in a diamond-shaped pattern of leaf scars, the marks left by the leaves that had already fallen, giving the entire trunk the appearance of a reptile's scales. Modern trees grow by adding rings of wood through a lateral meristem, building girth over decades. Lepidodendron grew almost entirely upward for most of its life, remaining essentially unbranched until it reached near its full height, then forking once, twice, and finally producing a crown of photosynthesizing leaves and a single massive cone of reproductive material before dying. Its entire lifespan was approximately 15 years. The Carboniferous coal forest was a landscape of enormous, fast-growing, short-lived towers of photosynthesizing tissue, not the long-lived forest giants of the modern world. Beneath the Lycopsid canopy, the ground was wet. The Carboniferous was a period of extensive tropical swamps across the equatorial regions of Laurussia, the northern portion of the assembling Pangaea, and the water table was at or near the surface for most of the year. The smell would have been extraordinary. The decomposing organic material of fallen Lycopsid trunks accumulating in the anaerobic mud, unable to rot completely because the microorganisms capable of decomposing lignin, the structural polymer that gives wood its rigidity, had not yet fully evolved. This is the origin of coal. The Carboniferous coal deposits that powered the Industrial Revolution are the compressed, transformed remnants of this undecomposed wood, the physical legacy of a forest ecosystem in which the trees fell faster than anything could break them down. In this wet, oxygen-rich ancient forest, the giant arthropods ruled. Meganeura patrolled the canopy at tree crown level, its 70-centimeter wingspan carrying it between the forking Lycopsid tops in search of smaller insects and possibly small vertebrates. Below, Arthropleura moved through the understory on its hundreds of legs, the 2.5-meter millipede relative navigating between the bases of the enormous trunks. The forest floor was a community of early arachnids, scorpions, harvestmen, early spiders, and of the small, four-legged amphibians that had colonized the land from the Devonian rivers, and that were, in the Carboniferous, still dependent on water for reproduction, returning to the swamps to lay their eggs in the wet season. It was in this world that the amniote egg was invented. We do not know exactly when or exactly by which lineage, but the fossil record of early amniotes, Hylonomus in Nova Scotia, Archaeothyris in the same Coal Age deposits, places the first egg-laying water-independent vertebrates firmly in the Carboniferous coal swamp environment. The invention was transformative not because it immediately changed the world, but because it removed a constraint that had limited terrestrial vertebrates to the vicinity of water for the entire previous history of their existence on land. The amniote could walk away from the swamp and never come back. It could climb the upland ridges above the coal forest and colonize the dry, seasonally arid environments that amphibians could not tolerate. It could, in principle, go anywhere that food and shelter existed, unconstrained by the need to keep its eggs wet. From the single amniote ancestor, the two lineages that would eventually produce all land vertebrates, synapsids and sauropsids, diverged in the late Carboniferous. The divergence is recorded in the skull. The sauropsid skull has either two temporal openings behind the eye or none at all, while the synapsid skull has exactly one. This single anatomical difference, modest in appearance, marks one of the most consequential bifurcations in the history of vertebrate evolution. The sauropsid line, over the following 300 million years, would produce the lizards and snakes, the turtles, the crocodilians, the pterosaurs, the dinosaurs, and the birds. The synapsid line would produce Dimetrodon, the gorgonopsians, the cynodonts, the first mammals, and, eventually, every warm-blooded animal alive today, including every person who has ever existed. The early Permian world in which Dimetrodon lived was dramatically different from the Carboniferous coal swamp world in which its amniote ancestors had first appeared. The assembly of Pangaea was changing the global climate. The closing of the seaways between the colliding continents was disrupting the ocean circulation patterns that had kept the Carboniferous tropics warm and wet, and the interior of the growing supercontinent was experiencing the continental climate extremes. Hot summers, cold winters, seasonal aridity that always characterize the interiors of very large landmasses. The coal forests were retreating, replaced by conifer forests and open, drier landscapes. And in these more open, seasonally dry environments, the advantages of the amniote body plan were fully expressed for the first time. Dimetrodon dominated the early Permian food web of what is now the southern United States and Western Europe, regions that were then equatorial in position, located near the center of the Pangaea landmass at low latitudes. The red beds of Texas, the Permian sedimentary formations in which most North American Dimetrodon fossils are found, preserve a community of remarkable ecological completeness. Multiple Dimetrodon species of different sizes filling different predator niches, Edaphosaurus as the sail-backed herbivore counterpart, and a diversity of smaller synapsids occupying the smaller predator and herbivore roles. This community, preserved in the iron-rich red sediments of an ancient river floodplain, is one of the most complete records of a pre-dinosaurian terrestrial ecosystem available anywhere in the world. And then the therapsids arrived, and the world changed again. The therapsid radiation of the Middle and Late Permian was the biological equivalent of the Cambrian explosion for terrestrial vertebrates. An explosive diversification of body plans, ecological roles, and physiological approaches that produced, within approximately 30 million years, the full range of large vertebrate ecological guilds that would characterize terrestrial communities until the modern day. Large herbivores, large predators, medium-sized omnivores, small insectivores, burrowing forms, aquatic forms. Every ecological role that large terrestrial vertebrates can occupy was filled by a therapsid during the Permian, with a sophistication of biological design that had not previously been achieved on land. The dicynodonts deserve more attention than they typically receive. In the popular imagination of the pre-dinosaurian world, the predators dominate: Dimetrodon, the gorgonopsians, and the herbivores are afterthoughts. But the dicynodonts were, by any ecological measure, the most successful large herbivores that the Paleozoic produced. At their peak diversity in the mid to late Permian, they occupied an enormous range of body sizes, from small burrowing forms the size of a rat to large browsers comparable to modern cows, and a corresponding range of ecological roles. Their unique skull plan, the reduced or absent teeth replaced by a keratinous beak, combined with the persistent upper tusks in most species, gave them a feeding apparatus of remarkable versatility. The beak could crop vegetation with precision. The tusks could be used for digging tubers and roots in hard soil. The jaw mechanics allowed a shearing bite that could process tough plant material more efficiently than the simple puncture and crush bite of most early herbivores. Inostrancevia was the apex of the gorgonopsian radiation, and its biology invites comparison with any predator that has ever lived. Standing approximately 1.5 meters at the hip, with a skull length of 45 centimeters and upper canines projecting 15 centimeters below the lower jaw when the mouth was closed, Inostrancevia was a predator built to bring down large prey in a single decisive attack. The canines were not simply large teeth. They were precision instruments, laterally compressed and serrated on both edges in some species, designed to penetrate deeply and then shear through flesh and blood vessels with a single closing of the jaw. Research on gorgonopsid tooth microstructure, published through 2025, has shown that their enamel had a specific layered structure that prevented tooth fracture during the enormous stresses of prey capture. The same general solution to tooth durability that was independently evolved by the saber-toothed cats of the Pleistocene, and that, in a modified form, is present in your own teeth. The behavioral ecology of the gorgonopsians can be partially reconstructed from their anatomy and from comparison with modern predators of similar build. The more erect hindlimb posture, the limbs held closer to beneath the body than the full sprawl of modern lizards, gave gorgonopsians a more efficient gait, and probably the capacity for sustained locomotion that sprawling-limbed predators lack. The temporal opening of the skull had enlarged to accommodate powerful jaw-closing muscles. The secondary palate, partially developed in some gorgonopsians, allowed breathing and swallowing to occur simultaneously, reducing the vulnerability of a predator in the act of consuming large prey. These are the biological features of an active, alert, physiologically capable predator, not the sluggish, slow metabolism opportunist that popular images of pre-mammalian synapsids sometimes suggest. The therocephalians were severely reduced, but not eliminated. The cynodonts, the group from which mammals would descend, survived in what appears to have been very small numbers, their survival in the early Triassic being one of the most consequential coincidences in the history of vertebrate evolution. The world that emerged from the end Permian extinction was, for millions of years, a biological wasteland. The early Triassic, the 3 to 5 million years immediately following the extinction, is characterized in the fossil record by what paleontologists call the coal gap, a period in which coal deposits, which require the accumulation of plant material in waterlogged environments and thus require productive forest ecosystems, are almost entirely absent from the geological record. The forests of the Permian were gone. The brachiopod-dominated seafloors were gone. The fusulinid foraminifera were gone. They never recovered, their entire group being one of the complete casualties of the extinction. The reef ecosystems did not return for approximately 10 million years after the extinction. Lystrosaurus inherited this empty world. The dicynodont survivor of the end Permian extinction was, by the standards of the Permian, an unimpressive animal, a medium-sized, barrel-bodied herbivore with a beak and 2 tusks. Approximately 1 meter long in the common early Triassic species. with a sprawling posture and a probable biology not dramatically different from that of a modern pig. But in the early Triassic, Lystrosaurus was the dominant vertebrate on land. Its fossils are found on every continent that was part of Pangaea at the time: Antarctica, Africa, India, Asia, Europe, in numbers that constitute a percentage of the total vertebrate fossil record, unprecedented for any single large vertebrate species before or since. Lystrosaurus was not ecologically diverse. It was ecologically monopolistic, the last herbivore standing in a world that had lost every other large land animal, filling the herbivore role by sheer default. The recovery of terrestrial vertebrate diversity after the end Permian extinction took approximately 10 million years, a geological interval that is short in the context of the history of life, but represents an enormous span of biological time in terms of generations and evolutionary opportunity. Through the early and middle Triassic, the therapsid survivors, primarily dicynodonts and cynodonts, diversified and recovered some of the ecological complexity that the Permian had achieved. New carnivorous cynodonts evolved to replace the lost gorgonopsians. New dicynodont herbivores diversified from the Lystrosaurus stock to fill the herbivore niches that the extinction had vacated. And in the Middle Triassic, the first true mammals, tiny, shrew-like animals with the full mammalian dentition and the three-bone middle ear, appeared in the fossil record. But the therapsids did not reclaim their Permian dominance. The reason is the archosaurs. The archosaurs, a group of diapsid reptiles that includes the ancestors of both dinosaurs and modern crocodilians, had survived the end Permian extinction in a marginal role, their diversity limited and their ecological significance minimal compared to the dominant therapsids. But the early Triassic was, in terms of global climate, a world for which the archosaur body plan was better suited than the therapsid body plan. The early Triassic was hot, very hot, by the standards of any period in the subsequent history of the Earth, and the tropical and subtropical regions of Pangaea experienced temperatures that challenged even the most drought-resistant organisms. The erect, more efficient locomotion of archosaurs gave them advantages in the energetically demanding hot environments of the early Triassic tropics. The archosaur lung, which operated on a flow-through principle, similar to modern birds rather than the bellows-style lung of mammals and most other vertebrates, may have been more efficient in the high-temperature, potentially oxygen-depleted conditions of the earliest Triassic. Through the Middle and Late Triassic, the archosaurs diversified rapidly while the therapsids declined. The large herbivore role, which dicynodonts had dominated in the early Triassic, was progressively taken over by increasingly large archosaurian herbivores. First, the attosaurs, armored plant-eating archosaurs, and then the sauropodomorph dinosaurs. The large predator role, which cynodont therapsids had partially reclaimed after the Permian extinction, was taken over by archosaurian predators, first the Rauisuchians and then the theropod dinosaurs. By the end of the Triassic, the therapsids had been reduced to the tiny, nocturnal, insect-eating creatures that would carry the mammalian lineage through 200 million years of dinosaur dominance. the ancestors of every mammal alive today, surviving in the margins of a world that the dinosaurs now owned. The end Triassic mass extinction, a second global catastrophe caused by a second large igneous province eruption, this one associated with the opening of the Atlantic Ocean as Pangaea began to fragment, eliminated many of the remaining Archosaurian groups and the remaining large therapsids, clearing the ecological space into which the dinosaurs subsequently radiated with such explosive success. Our ancestors responded to this second catastrophe the same way they had responded to the first, by becoming smaller, by becoming more nocturnal, by becoming more flexible in their diet and behavior, by retreating into the ecological margins and waiting. The waiting would last 200 million years, but it would eventually end when another asteroid struck and the long dominance of the dinosaurs was terminated in a day. The story of how the archosaurs displaced our ancestors and of what those ancestors endured during the 200 million years that followed deserves to be told in full, because it is the most consequential period of waiting in the history of life. The Triassic takeover was not sudden. It was gradual, opportunistic, and driven by a combination of factors that made the post-extinction world better suited to the archosaur body plan than to the therapsid body plan. The key advantage was locomotion. Early archosaurs had evolved a more erect hind limb posture than most of their contemporaries. The hind limbs held directly beneath the body in a parasagittal plane, rather than extending outward from the body, as in the sprawling gait of lizards and early therapsids. This erect posture allowed longer strides, more efficient locomotion, and, crucially, the ability to breathe while running without the constraint that afflicts sprawling-limbed animals. When a lizard runs, its body bends from side to side, compressing first one lung and then the other, making sustained running and breathing simultaneously difficult. An erect-limbed animal has no such constraint. It can run indefinitely. Euparkeria capensis, a small, agile archosaur from the early Triassic of South Africa, known from beautifully preserved fossils in the Karoo Basin, represents something close to the ancestral condition of the archosaur lineage from which both dinosaurs and crocodilians descended. It was approximately 50 centimeters long, lightly built, with hind limbs somewhat longer than its forelimbs, suggesting the capacity for occasional bipedal sprinting. Its teeth were sharp and recurved, its eyes large, its overall aspect suggesting an active, fast-moving insectivore or small vertebrate predator. Nothing about Euparkeria suggested that its descendants would rule the Earth for 180 million years. But the biomechanical advantages encoded in its skeleton, the improved locomotion, the more efficient respiration, the ankle joint architecture that would eventually allow the fully erect stance of the dinosaurs, were already present in miniature. Through the Middle Triassic, the archosaurs diversified into the ecological roles that the Permian extinction had emptied and that the therapsids had been struggling to refill. The Rawisuchians, large, erect-limbed archosaurian predators, began to displace the carnivorous cynodonts from the large predator role. The aetosaurs, heavily armored, plant-eating archosaurs with bodies like small tanks, moved into herbivore niches that dicynodonts had previously dominated. And the dinosaurs themselves first appeared in the fossil record of Argentina in rocks approximately 230 million years old, the Ischigualasto Formation of the Carnian Age, as small, bipedal, fast-moving animals that in their initial appearance were no more ecologically dominant than dozens of other vertebrate groups present in the same deposit. Eoraptor lunensis, one of the earliest dinosaurs, was approximately 1 meter long and weighed perhaps 5 kilograms, the size of a small dog. It was a generalist predator or omnivore, its teeth showing the mixed morphology of an animal that ate both small vertebrates and plant material. Nothing about Eoraptor suggested the ecological dominance that its lineage would eventually achieve. But the dinosaur body plan, the fully erect hind limbs, the lightweight skeleton, the efficient respiratory system inherited from archosaur ancestors was already established, and the end Triassic mass extinction that occurred approximately 201 million years ago would remove the last major competitors and open the world to the dinosaurian radiation that defined the Jurassic and Cretaceous. The directional trend is visible in the fossil record across the Triassic, cynodont body sizes declining generation by generation. This miniaturization was not a defeat. It was a survival strategy of extraordinary effectiveness. The first true mammals, animals with the complete mammalian dentition, the fully mammalian middle ear with its 3 tiny bones, and the mammalian ankle and skull structure, appear in the fossil record approximately 225 million years ago, in the late Triassic. Morganucodon watsoni, known from fossils found in limestone fissure deposits in Wales and in equivalent aged rocks in China, South Africa, and North America, is one of the best known of these earliest true mammals. It was approximately 10 centimeters long, the size of a modern shrew, with a pointed snout, large eye sockets suggesting large eyes adapted for low-light conditions, and teeth worn in a pattern consistent with an insectivorous diet. Morganucodon was nocturnal, small, fast-breeding, and energetically expensive to maintain. A warm-blooded insectivore in a world of cold-blooded giants, surviving by exploiting the nighttime ecological space that the dinosaurs, whose visual systems were primarily adapted for daylight, could not efficiently occupy. The nocturnal bottleneck, the long period during which the ancestors of all modern mammals were restricted to small body sizes and nocturnal activity by the ecological dominance of the dinosaurs, has left traces in mammalian biology that persist to the present day. The color vision of most mammals is inferior to that of birds and reptiles, because the early mammals lost the photoreceptor types needed for full color vision during the tens of millions of years in which they lived in darkness. Only the primates, which re-evolved enhanced color vision relatively recently in evolutionary time, have partially recovered the full color perception that our synapsid ancestors possessed before the dinosaurs forced them into the night. The large, forward-facing eyes of primates, eyes that give excellent night vision and depth perception, but sacrifice the wide field of view that prey animals require, are adaptations that developed in small, nocturnal, branch-navigating mammals, and that were subsequently elaborated in the primate lineage into the visual system that eventually gave our ancestors the ability to make and use tools with precision. For 160 million years, our ancestors lived as small, nocturnal, mostly inconspicuous animals in a world owned by dinosaurs. They lived in the leaf litter. They lived in burrows. They lived in the trees. In some lineages, developing the grasping hands and enhanced visual systems that would eventually characterize the primates. They were not passive victims of dinosaurian dominance. The Mesozoic mammals were ecologically diverse and biologically sophisticated, some lineages achieving body sizes larger than rats, some exploiting aquatic environments, some gliding, some burrowing with extraordinary specialization. The Jurassic and Cretaceous were not a mammalian dark age, but a mammalian incubation, a period during which the fundamental mammalian body plan was refined, tested, and diversified in the ecological margins, preparing for the moment when the constraints of dinosaurian dominance would be removed. That moment came 66 million years ago, when the Chicxulub asteroid struck the Yucatan Peninsula and changed everything in a geological instant. The bolide impact released energy equivalent to billions of nuclear weapons, triggering wildfires, earthquakes, tsunamis, and a stratospheric debris cloud that blocked sunlight globally for months to years. The non-avian dinosaurs, the ecological dominance of every terrestrial ecosystem on Earth for 160 million years, were eliminated. The large terrestrial ecological space was vacated in a single catastrophic event. And for the first time since the Triassic, the mammals were no longer constrained by the presence of large, active, warm-blooded competitors in the daylight ecological world. The Paleocene and Eocene explosions of mammalian diversity that followed the end Cretaceous extinction are the direct consequence of 160 million years of enforced constraint suddenly being removed. The mammals did not need to invent new biology for the post-dinosaur world. They already had warm blood, differentiated teeth, advanced brains, flexible limbs, and complex social behaviors developed over the previous 200 million years of synapsid evolution. They needed only to grow larger and diversify into the ecological roles that the dinosaurs had vacated. Within 10 million years of the end Cretaceous extinction, the mammals had produced animals the size of modern rhinoceroses. Within 20 million years, the first whales had entered the ocean. Within 66 million years, one mammalian lineage had developed the neural complexity to understand that the warm blood in its veins, the bones in its ears, and the teeth in its skull were the legacy of a world that existed before the dinosaurs, endured through the dinosaurs, and survived everything the history of the Earth had thrown at it. The 200 million year wait was over. The world before the dinosaurs had finally become the world after them. The biological legacy of that world, the specific features that connect Permian therapsids to the person reading these words, deserves to be laid out in precise anatomical detail. The world before dinosaurs is not a world of lesser animals waiting to be replaced by something better. It is a world of biological achievement, whose scale and sophistication was not surpassed by anything in the subsequent history of vertebrate life, until the appearance of the mammals in the Cenozoic. The gorgonopsians were not inferior to the theropod dinosaurs that replaced them. They were different, synapsid rather than sauropsid in their fundamental architecture, and they were eliminated not because of any biological inadequacy, but because a volcanic eruption in Siberia released enough greenhouse gas to kill 96% of the species on Earth. 100 million years of evolutionary achievement erased, not reduced, not damaged, erased. And yet the chain did not break. And yet from that near zero, life recovered. From the single surviving cynodont lineage and the small, unimpressive cynodont survivors. The therapsid heritage persisted through the catastrophe and eventually produced, 200 million years later, every mammal on Earth, every whale and bat and elephant and human being. The chain of continuity that links Dimetrodon to you is unbroken, despite everything that intervened. It runs through the Permian extinction, through the Triassic marginalization, through 200 million years of dinosaur dominance, through the tiny nocturnal insectivores that carried the mammalian program through the Mesozoic in the ecological shadows. It runs, without interruption, from the coal swamps of the Carboniferous to the moment you are reading these words. The world before dinosaurs was our world. It was the world that made us possible by making the biological innovations, the synapsid skull, the differentiated teeth, the warm blood, the amniote egg, that are still the foundation of our biology today. The dinosaurs have their movies and their museums and their cultural dominance. The specificity of what we inherited from the pre-dinosaurian world deserves more than passing mention because it is not merely metaphorical. The biological legacy of the synapsid Permian is not a matter of vague evolutionary ancestry. It is a set of specific, identifiable anatomical and physiological features that are present in your body right now and that can be traced with precision to their origin in specific Permian and Carboniferous synapsids. Your teeth are the most immediately visible of these legacies. The human dentition, incisors for biting, canines for piercing, premolars and molars for chewing, is a specifically mammalian feature that appears in the fossil record for the first time in the paleosaurs of the early Permian. No living reptile has this configuration. No dinosaur had it. The differentiation of tooth types in response to different functional demands, different teeth doing different jobs within the same jaw, is a synapsid innovation that Dimetrodon was already exploiting 300 million years ago. When you bite an apple with your incisors and chew it with your molars, you are performing a biomechanical sequence that was invented by a sail-backed predator in an early Permian Texas River floodplain. The bones of your middle ear are perhaps the most extraordinary example of evolutionary repurposing in the history of vertebrate life, and they connect you directly to the therapsid experimentation of the Permian. In reptiles, and in early synapsids like Dimetrodon, the lower jaw is composed of multiple bones. The dentary, which bears the teeth, and several smaller bones. The articular, the quadrate, and others, that form the jaw joint and transmit sound vibrations to the inner ear through a single bone called the stapes. In mammals, the lower jaw is composed of a single bone, the dentary, and the former jaw joint bones have migrated into the middle ear, where they function as the malleus and incus. two of the three tiny bones that amplify and transmit sound vibrations to the inner ear. This transition can be traced in exquisite detail through the cynodont fossil record of the Triassic, where intermediate forms show the gradual reduction of the non-dentary lower jaw bones and their progressive incorporation into the auditory system. Your ability to hear the range of frequencies that human speech occupies, the physical basis of language, of music, of every meaningful sound you have ever perceived, depends on bones that were once part of the jaw of a Permian therapsid. Your thermoregulation, the ability to maintain a constant body temperature of approximately 37 degrees Celsius, regardless of whether the external temperature is -40 or +40, is the metabolic legacy of the therapsid revolution. Modern reptiles, including the descendants of the dinosaurs, are ectothermic. Their body temperature tracks the environmental temperature, and their activity level rises and falls with it. Mammals are endothermic. They generate body heat through elevated metabolic activity, burning food at rates 4 to 10 times higher per unit body weight than equivalent-sized reptiles. This metabolic investment is expensive. It is why you need to eat every day, and a crocodile of the same body weight can go weeks between meals. But it provides the ability to be active in cold weather, to maintain cognitive function across a wide range of environmental temperatures, and to sustain high levels of physical activity indefinitely, rather than in short, exhausting bursts. The endothermic metabolism that enables everything you do was developed in the Permian therapsids, probably beginning with the earliest therapsids and reaching full mammalian levels in the cynodonts of the Triassic. Your jaw musculature, the masseter, the temporalis, the pterygoid muscles that allow you to chew food into small pieces rather than simply swallowing large chunks, is the product of the expansion of the temporal opening in the synapsid skull that began with the very first synapsids in the Carboniferous. The temporal opening created space for larger jaw muscles. Larger jaw muscles allowed more powerful and precise chewing. More powerful chewing allowed the exploitation of harder and more nutritious food items, seeds, roots, hard-shelled invertebrates that less well-equipped herbivores and omnivores could not access. The expansion of the synapsid temporal opening over the course of the Permian and Triassic, traceable in exquisite detail through the fossil record, is the anatomical history of the machinery that allows you to eat. Your facial expressiveness, the ability to communicate emotion, intention, and social information through the movement of the muscles of your face, is a mammalian feature that has no equivalent in reptiles or birds. And its origin lies in the vibrissae, the sensory whiskers that some therocephalians already possessed in the Permian. The facial muscles that move whiskers in modern mammals were the precursors of the facial muscles that produce human expression. The capacity for nuanced facial communication that is so central to human social life, the raised eyebrow, the smile, the frown, is the elaborated product of a sensory system that some late Permian therapsids were already developing in the Pangaean uplands. Your brain, or more precisely, the limbic system, the neural architecture underlying emotion, memory, and social bonding, is a mammalian structure that has no precise equivalent in the reptilian brain. The emotional life of a mammal, with its capacity for attachment, for grief, for play, For the sustained social bonds that underlie all human society is a product of the mammalian neural elaboration that began in the synapsid lineage and reached its full expression in the early true mammals of the Triassic. Whether the gorgonopsians had something resembling emotions, whether Inostrancavia experienced something when its cubs were threatened, cannot be answered from the fossil record. But the neural machinery that makes your emotional life possible is traceable to the synapsid innovations of the Permian. And the gorgonopsians were close enough to that ancestry that the question is not absurd. The world before dinosaurs is the world that made you. Every feature of your body that distinguishes you from a reptile, your warm blood, your differentiated teeth, your middle ear bones, your jaw muscles, your facial expressiveness, your emotional brain, was developed in the Permian and Carboniferous by animals that have been dead for 250 million years. The dinosaurs had nothing to do with any of it. They inherited a world already shaped by 100 million years of synapsid innovation, occupied the ecological roles that the Permian extinction had cleared for them, dominated the planet for their allotted time, and were then eliminated by an asteroid impact that opened the door for the mammals that had been waiting in the ecological margins since the Triassic. Stand in front of a mirror and look at your face, the differentiated teeth visible when you smile, the jaw that moves sideways and forward and backward, not just up and down like a reptile's jaw when you chew, the expressiveness that plays across the muscles of your face in response to whatever you are feeling. All of it, every feature of that face, was invented before the first dinosaur ever walked the earth. All of it was paid for with the lives of the gorgonopsians and the cynodonts and the therocephalians that built it over 100 million years, and then lost everything to a volcanic catastrophe that reset the planet. You exist because a few small, nocturnal, insectivorous cynodonts survived the end of their world and carried the biological program forward through 200 million years of darkness. The world before dinosaurs did not end. It became you. The world before them has only the fossil record and the bodies that carry its legacy forward. Your body, which is built on the biological architecture of a world that ended 252 million years ago, and that, in a very real sense, has never entirely ended, because the lineage it created is still here.